Hello everyone. My name is Jakub Chwastek and I am postdoc in Witek Konopka's lab. We are part of the Polish Center for Technology Development in Łukasiewicz Network in Poland, and our institution is located in Wrocław. Today I will present you a paper published by Ramirez and co-operators in Cell Metabolism in 2022. And its title is Hypothalamic pregnenolone mediated recognition memory in the context of metabolic disorders. I would like to start by introducing you the main topic of this article, which is obesity, and related to that metabolic disorders and cognitive changes. As you may know, it is one of the biggest problems of the modern world, and according to the World Health Organization, in 2022, in the European region, overweight and obese will affect almost 60% of adults, and then this number tripled since 1975. Big belly may or may not be aesthetically pleasing for somebody's, but unfortunately is also major risk factor for diseases such as diabetes, hypertension, osteoarthritis, or cardiovascular disease, which are serious and one of the main problems for modern medicine. The brain main centers for regulating appetite are located in the arcuate nucleus, which is part of the mediobasal hypothalamus, and the best known for now populations of neurons that control food intake are AGRP and POMC neurons, which respond to central and peripheral signals about the body energy balance. And previous studies have shown that obesity may associated with mild cognitive impairment, and cholesterol metabolism has been also suggested as a possible link between metabolic and cognitive disorders. So let me go back to the graphical abstract. And as you can see, the authors have shown in paper that disturbances in metabolism, mitochondrial metabolism of cholesterol can cause impairment in cognitive functions, which is observed both in obesity and in acute unhealthy diets. So they started with modeling animals, with feeding them for 4 days with a Western diet, whose energy profile was 40% kilocalories from fat and 43% from carbohydrates, or standard CHO diet, where the kilocalories from fat was much lower than in the experimental group. So Western diet was some kind of unhealthy diet observed in our modern society. As you can see, this short-term feeding with unhealthy Western diet did not cause any change in phenotype of the which were measured by body weight changes or blood glucose levels. In order to assess changes in cognitive functions in the animals, the authors used the Barnes-Maze test, BMT in short, which tests hippocampal dependent spatial memory. The animals were placed in an arena with some external cues, which you can see on the scan, and their task was to find an escape hole where other holes were closed. The mice were trained for 5 days, after which they rest for 2 additional days, and on the 8th day was the test day. and every hole in this test was closed. During the test day, parameters such as latency to the target hole uh, in seconds, time in the target quadrant, which is this one, and total distance were measured. And authors used also a second behavioral test, which is a novel object recognition test. Animals were trained after habituation, and this training session lasted 2 hours. And after that, object was replaced with another one. The parameters which were measured was exploration time, total distance, and discrimination index, which allows authors comparison of the time spent by animals exploring novel object in a simpler way than just values expressed in seconds. So authors with this test found that short-term feeding with Western diet did not alter animal spatial memory, but significantly impaired their recognition memory as measured in a novel object recognition test. Authors try to somehow explain these findings, so they try to examine hippocampal LTP, so long-term potentiation, which is the most widely proposed mechanism for memory storage. As you know, the hippocampus is a brain region crucial for the formation, organization, and storage of memories. The LTP process allows synapses to become stronger, which is probably the most important cellular process observed during learning and memory formation. And one of the most widely used tools for LTP analysis is theta burst stimulation, TBS in short, which mimics the natural firing pattern of hippocampal neurons. During this procedure, neurons are stimulated with repeated bursts at frequency of about 5 Hz, which allows LTP to be induced. So authors recorded the activity of hippocampal neurons in slices isolated from animals fed standard or short-term Western diet. And as you can see on the graph here and here, unhealthy diet impaired hippocampal LTP, which may be cause of the mild cognitive changes observed on the previous slide. Next, they measured the concentration of pregnenolone, which is a product of the cholesterol metabolism in the mitochondria, and it is a substrate for the synthesis of other steroids. Pregnenolone levels were measured in 3 different regions of the brain. 2 of them, the perirhinal cortex and the hippocampus, are involved in memory formation, and the Acute, as I said in the introduction, is a center that controls energy balance. And the authors find that the concentration of pregnenolone was reduced only in the Acute in animals fed with Western diet compared to the control group. Next, authors investigated the role of pregnenolone in cognitive functions. They canned animals intracerebroventricularly, ICV in short, and after habituation, after surgery, mice performed not training session, and after 2 hours before the test, 10 micrograms per mouse of pregnenolone or vehicle when injected. It is interesting that this kind of acute supplementation reverses the impairment of cognitive functions observed in animals fed with Western diet. For the next experiments, animals with a knockout of the STAR1 protein limited to the AGRP or POMC neurons were used, and authors confirm STAR-D1 knockout with PCR method in the arcuate and mediobasal hypothalamus in the AGRP STAR-KO line, and in the arcuate and pituitary in the POMC STAR-KO mice line. Additionally, they showed downregulation of STAR-D1 gene expression in targeted neuronal populations, and they checked the colocalization of STAR-D1 protein with fluorescent in situ hybridization in AGRP or POMC neuron populations. What's important, both mutant lines did not differ from the control group in metabolic parameters such as body weight, daily food intake, or blood glucose levels. As in an experiment with Western diet feeding mice, pregnenolone level was measured in the perirhinal cortex in both lines, hippocampus and arcuate. And interestingly, the level of pregnenolone was significantly reduced only in the arcuate of POMC STAR-KO line. And this change correlates with the cognitive impairments observed in animals fed Western diet for diets and was not present in AGR STAR-KO mice. And as I said, it was in the POMC STAR-KO mice. That's why this mutant line were used in the next steps of the experiments published in this paper. POMC STAR-KO mice phenotype did not differ from control mice in overall activity, depression-like behavior measured with the Thai suspension tests, or anxiety level test with 4 different tests such as open field, dark light box, elevated plus maze, or Marble Baring tests. Authors confirm cognitive dysfunction of POMC STAR-KO line with additional test, novel odor recognition, which is pretty similar to NORD. The test consisted of 3 phases: habituation, training, and test phase. In ARENA, there was available 2 identical leaky containers which were placed in the cage or arena. During the training phase, the mice had access to 2 identical non-social odors, which were some diet-related odors. In the test phase, one of odors were replaced by a new non-social odor, so some new diet for animal. And parameters such as exploration time and total distance were measured. And discrimination index was calculated by the author based on this previous mentioned parameters. And as you can see, cognitive functions of the POMC neurons in this test were significantly impaired compared to the control animals, such as in the NOR test. Previous studies have shown that cognitive impairment may be associated with central insulin resistance. So the authors investigate the state of insulin signaling pathway in POMC star KO mice. In this purpose, they administered 5 units of insulin via the inferior cava vein to anesthetized mice, which were fasted overnight. And they isolated different tissues and at different time points. They measured phosphorylation of AKT protein, which is one of the marker of the activity of the insulin pathway, intracellular insulin pathway. And authors found that in the Acute nucleus of the POMC star KO mice, this pathway was blunted or inhibited, but they did not observe this kind of changes in other tissues, such as other brain samples or skeletal muscles or livers. So they also used another one genetic model, and they generate POMC in an insulin receptor KO line, and they checked pregnenolone in the perirhinal cortex, hippocampus, and in arcuate. And they did not observe any significant changes in these parameters. The authors also show that insulin signaling in POMC neurons doesn't mediate cognitive performance, which they demonstrated using the open field test, BMT, and NOR. On the next step, authors confirm POMC neuron projections to the hippocampal subiculum, and they use for that injection to the arcuate AAV vector expressing DT tomato in the cytoplasm of POMC neurons, and green fluorescence protein presynaptically. And the green fluorescence was observed in hippocampal slices as shown here. And in this way, they confirmed these projections to this region. Moreover, hippocampal LTP levels were measured by authors, and they showed that POMC star KO mice LTP level in the slides were lower. And acute injection of pregnenolone, it least partially reversed this phenomenon. In addition, acute pregnenolone administration restores impaired cognitive functions in POMC star KO mice. The activity of POMC neurons in wild-type animals was also increased after injection of pregnenolone, and it was assessed by counting false positive neurons 90 minutes after injection of pregnenolone. As you can see, the number of of false positive cells was significantly higher than in the vehicle injected group. Moreover, this parameter didn't change in other population of the neurons than POMCs. The role of POMC neurons activity was also investigating using designer receptors exclusively activated by the Zarney drug system, DREAD in short. And as you know, it is based on artificial G protein coupled inhibitory or activatory receptors, which in this case were introduced to POMC neurons population via a V vector, injected bilaterally to arcuate of control animals, which was POMC CRE plus, or to the research group, which is POMC star KIO group. Additionally, administration of specific agonist for artificial receptors, which called CNO, provides to action of these artificial receptors, so activation of or inhibition of them. This experiment showed that enhancement of cognitive function in POMC star KIO mice caused by EVC pregnenolone injection is blocked when POMC neurons are pharmacologically inhibited. In addition, inhibition of POMC neurons blunts also cognitive functions in the control animals. And this state is not observed in active POMC neurons groups. Authors also induced metabolic disorders in animals by feeding them with Western diet for 16 weeks. They observed significant increase in body weight and blood glucose levels. In the BMT, obese animals spent more time in the target quadrant on the day of the test, and the authors were unable to explain these results. The Western diet did not alter the behavior of the mice in the open field test when they compared to control. And what's important, the data from North are similar to previous observed results. And obese animals' cognitive functions were impaired when measured with this test, with North. And additionally, authors checked the pregnenolone level, which decreased in arcuate of Western diet fed animals. And this part was the last experiment with animals. So the authors did not check whether pregnenolone injection improved cognitive functions in these animals. In the last part of the article, the authors used a cohort of metabolically healthy MHO or unhealthy, so MUO, obese patients. They collect from patients CSF and plasma to check their pregnenolone levels and clinical parameter respectively. The cognitive status of the patients was measured using the mini mental status examination, MMSE, which is commonly used screening test to assess cognitive impairments in adults. I have included a sample test, and as you will check these questions, you can see that there are different tasks which assess different abilities of the patients. Blood analysis showed significant differences between MHO and unhealthy patient groups in diabetes-related parameters, such as fasting glycemia, insulin resistance, glycosylated hemoglobin, so HbA1c, and HDL cholesterol, but not in MMSE score or cerebrospinal fluid pregnenolone levels. But authors also check the correlation of pregnenolone level with other parameters, and they found that it is negatively correlated with BMI and positively correlate with MMSE score in metabolically unhealthy group, but not in the healthy control. So to summarize for this article, the author showed that even a short-term Western diet was sufficient to impair recognition memory, which was paralleled by pregnenolone levels in the RQ8, but not in the memory-related regions. In addition, neurosteroid signaling in POMC neurons mediates recognition memory. Human studies have shown that central pregnenolone levels may be relevant in cognitive impairment associated with metabolic disorders. And in the end, we can also find information about the limitations of the study. And the authors point out that pregnenolone is probably indirectly responsible for the change in cognitive function because it is the precursor for the biosynthesis of other neurosteroids. There is also information about possible alternative pathways regulated by the DREAD system. And finally, human CSF pregnenolone probably reflects general biochemical changes in the brain. So it is not very specific biomarker. Thank you for your attention, and I'm waiting for your opinion about this article, and I'm looking forward to our discussion next Thursday.